The overall goal of this procedure is to prepare and purify an N-tos-iodoaziridine. This is accomplished by first generating diiodomethyllithium by deprotonation of diiodomethane at low temperature and reacting with an imine. The second step is to rapidly warm the reaction mixture to promote cyclization of the diiodide intermediate to the desired iodoaziridine product. Next, the stability of the iodoaziridine product to various stationary phases used for chromatography is assessed to determine the most appropriate for purification. The final step is to purify the iodoaziridine and perform proton NMR analysis. Ultimately, this screen of stationary phases for chromatography is used to show that basic alumina modified to activity 4 is most appropriate for the purification of the iodoaziridine.
We first had the idea for this method when we found that the new iodoaziridine products were unstable to silica gel chromatography, and we required an alternative purification.
From here, we designed a procedure to assess the stability of the iodoaziridine to a range of stationary phases for chromatography, which can also be applied to other potentially sensitive compounds.
Demonstrating this procedure will be Tom and Dom, who are both PhD students in my research group.
First, flame dry a 100 ml round bottom flask containing a stir bar and fitted with a septum under a stream of argon. After allowing the flask to cool to room temperature, add 5.7 ml of anhydrous tetrahydrofuran and 2.7 ml of anhydrous diethyl ether via syringe, and 315 microliters of freshly distilled hexamethyl disilazane via a microsyringe. Place the flask in a suitably sized dewar that will